So in the previous section, we looked at the light reactions, where the photosystems embedded in the thylakoid membrane trapped energy from sunlight and stored them as ATP and NADPH. Now it's time to take the ATP and NADPH generated by the light reactions and use them to fuel the Calvin cycle, where we fix carbon in order to create sugar molecules. So in the light-independent reactions is where we see the carbon fixation, and carbon fixation occurs in the Calvin cycle. By fixing CO2 molecules, we're able to build carbohydrates that cells can then use as fuel. The energy is derived from the light-dependent reactions in the form of ATP through both cyclic and non-cyclic photophosphorylation. This energy that's captured from the sun is the energy that's going to drive the endergonic reaction of generating these carbohydrates. We also see that there's reduction potential because of the NADPH that we got from photosystem 1 during the light-dependent reactions. And just as we removed electrons and hydrogens to break the bonds in cellular respiration, here we're able to add electrons and hydrogens to these CO2 molecules in order to generate carbohydrates. Let's take a quick look back at the non-cyclic and cyclic photophosphorylation. This figure's a good one. Remember that photosystem 2 comes first, and it's P680. Here, electrons are excited. They move down through an electron transport chain in order to generate ATP. Once these electrons reach the reaction center in photosystem 1, they're excited, and they move through another electron transport chain and are finally picked up by NAD+. Now we've generated one ATP and one NADPH. You may recall that we need 3 ATP for every 2 NADPH, or 1.5 for each. And so this Z scheme, or non-cyclic photophosphorylation system, will run twice. Electrons are not moving in a cycle. They're leaving this photosystem and they're ending up in NADPH. Remember, the electrons here are replaced by splitting water. In order to generate an additional ATP, the Z scheme will run twice in non-cyclic electron flow. And then photosystem 1 has the capacity to do cyclic photophosphorylation. So the primary acceptor will just drop the electrons down into the electron transport chain that's developing ATP instead of NADPH. Here, the electrons return to exactly where they left, and thus we have cyclic electron flow. So again, two things. We have energy in the form of ATP and reduction potential because we have electrons and hydrogens stored in this NADPH molecule. So the Calvin cycle was named after a guy named Melvin Calvin, and it's also known as C3 photosynthesis, and that's because we form a 3-carbon molecule at the end of the Calvin cycle. 2 3-carbon molecules come together to form a 6-carbon molecule that we'll call glucose. The key step in the Calvin cycle is carbon fixation, or the attachment of CO2 to another molecule called RuBP. The enzyme that's used here is called rubisco for short, or rubulose bisphosphate carboxylase oxygenase. It's a long name, so you can see why we call it rubisco for short. So now let's take a look at the 3 phases of this cycle. In the first phase, we have carbon fixation. This is where we take carbon dioxides coming into the cycle and fix them onto a molecule called RuBP. Here we generate PGA, or 3-phosphoglycerate. You may recognize this from the first reactions of glycolysis, where we split glucose into the first 3-carbon molecule. It was also PGA. In the next phase of the cycle, we have reduction. During reduction, there's a series of reactions that reduce PGA by adding electrons and hydrogens to G3P. We won't worry about the intermediates. In the third phase of the cycle, we're doing regeneration of RuBp. So very similar to what happens in the Krebs cycle. We're simply rearranging these carbons in order to produce RuBp so that it can again pick up carbon dioxide and continue round the cycle. We'll be considering how the Calvin cycle can produce one molecule of glucose. Although the glucose is not produced directly by this cycle, a series of reactions and 6 molecules of CO2 will eventually produce one glucose molecule. So essentially, this cycle needs to go round 6 times in order to incorporate enough carbon to create one 6-carbon glucose. However, the first product from it is glyceraldehyde 3-phosphate, or G3P. Each of these are 3-carbon molecules, so it takes 2 3-carbon molecules to come together to form one glucose. So let's review that on a larger scale. Remember that the enzyme Rubisco, this is the one with the very complicated name that we shorthand for Rubisco, is out in the stroma, outside the thylakoid membranes, but inside the chloroplasts. Rubisco is the key to carbon fixation. In fact, it's one of the most prevalent enzymes on the planet. So 6 molecules of carbon dioxide will enter this cycle. We're going to track the carbons. We have a bench, again a workbench. It's a 5-carbon workbench. So we'll take 6 molecules of RuBp, which are 5 carbons, and we'll add 1 molecule of CO2, which only has 1 carbon. And so here, for an instant, we'll have 6 times 6 carbon molecules. These 6 carbon molecules very rapidly get turned into 12 3-carbon molecules. Then we have reduction, where 12 ATP are added, 12 NADPHs, which are carrying electrons and hydrogens, and thus 3-phosphoglycerate is transformed into glyceraldehyde 3-phosphate. It's a much higher energy molecule. But again, we have 12 molecules. We've got 12 3-carbon molecules. So we're tracking all the carbons here. We had 5 carbons plus 1 carbon times 6 molecules, which gave us, for a moment, 6 6-carbon molecules. Then we split those 6-carbon molecules each into 2, so that gave us 12 3-carbon molecules. We add some energy in the form of ATP. We add some electrons and hydrogens that we take from NADPH. And now we have 12 molecules of higher energy 3-carbon sugars. After all, we've dropped off all those electrons. Now, let's skip ahead for a moment. How many carbons in all do we have over here in the 6 molecules of RuBp? Well, they're 5 carbons each. So we have 6 times 5 carbons equals 30 carbons. So we have to recycle 30 carbons. At the bottom here, we have 12 molecules of 3 carbons. So we've got 12 times 3 carbons equals 36 carbons. So we can spare 6 carbons. And that's exactly what happens. So 6 carbons fall out. We end up with 2 molecules of G3P equaling 6 carbons, and these are removed from the cycle. So 36 minus 6 leaves us with the 30 carbons that we started with in RuBp. In order to regenerate RuBp, This is where we need the addition of the extra ATP molecules. So 6 ATP are necessary to regenerate. 12 ATP and 12 NADPH are necessary for the reduction phase. And you remember there was a 3-to-2 relationship where we needed 3 ATP for every 2 NADPH. This is where the extra ATP comes in. So in essence, you could say these extra ATP that we use for the regeneration of RuBP are generated by the cyclic portion of photophosphorylation, or cyclic electron flow. So let's watch a quick animation to see if it can help us clarify the Calvin cycle.
When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life, providing food and oxygen for the complex food webs on both land and in the oceans.
So let's see how we might diagram this. First of all, I've started with a circle to represent the cycle. I'm divided into 3 phases: our carbon fixing phase, the reduction phase, and then the regeneration phase, where we'll regenerate RuBP. I've started out with the 6 RuBP molecules. Each of these are 5 carbons. And to these 5 carbons, we're going to add CO2. In order to keep track of 1 whole glucose, I'm going to multiply everything by 6. So 6 CO2 enter the cycle, and they combine with the 6 RuBPs. Instantaneously, the enzyme we call Rubisco fixes the carbon onto each of these 5-carbon RuBPs to make 6 transient 6-carbon molecules, which, if we're tracking carbons, gives us a total of 36 carbons. But this state only exists for a very brief period while we're still attached to Rubisco. Each of these molecules are very quickly split. They become split into 12 3-carbon molecules, so still equals 36 carbons. This is the intermediate PGA. otherwise known as 3-phosphoglycerate. Now it's time for the reduction phase, in which place we're going to use the energy of ATP to fuel the addition of electrons and hydrogens. That is, reducing PGA eventually into G3P, which is glyceraldehyde 3-phosphate. So we have the addition of 12 ATP, which will drop off their phosphates and leave as 12 ADP. In addition, 12 NADPH will enter, drop off pairs of electrons and hydrogen ions, and leave as 12 NADP+. After the energy is supplied and the 3-carbon molecule becomes reduced, it's now called glyceraldehyde 3-phosphate, or G3P. And we have 12 of these. Again, to track the carbons, we'll have 12 times 3 carbon molecules. We've still got 36 carbons, which means we can afford to lose 2 G3Ps, which equals 6 carbons. Eventually, these can go on to form glucose. Now what we have is the regeneration phase. In order to regenerate, we need to have more ATP investment. There are 6 ATPs that are invested in the regeneration phase, which will leave as 6 ADPs. And after we've removed 6 carbons that can eventually be made into glucose, we'll be left with 30 carbons. These 30 carbons will be rearranged and regenerated into RuBP. 5 molecules, 6 molecules of 5-carbon RuBP. 6 times 5 equals 30. We've accounted for all of the carbons in this cycle. Once again, this diagram looks much nicer on my page than it does on this PowerPoint slide. My apologies for the messiness. So let's quickly look at the output of the Calvin cycle. Because as you may already have noticed, glucose is not the direct product of the Calvin cycle. G3P is the direct product, otherwise known as glyceraldehyde 3-phosphate. You only need to know it as G3P, but it's a 3-carbon sugar. It can be used to form sucrose, which is a major transport sugar that we see in plants, and is a disaccharide that's made from fructose and glucose phosphate. We could also use it to make starch, which is an insoluble glucose polymer. And as you've already learned, starch is a storage molecule for long strings of glucose phosphates. So photosynthesis uses the products of respiration as starting substrates, and respiration uses the products of photosynthesis as starting substrates. You might have noticed some relationship between these two. Even when we look at the equations, one appears to be the reverse of the other. The production of glucose from G3P even uses part of the ancient glycolytic pathway running in reverse. You'll recall that glucose is first split into G3P, and then we steal electrons from it. The principal proteins that are involved in the electron transport and ATP production in plants are evolutionarily related to those that we see in the mitochondria. For example, the proteins that we see in the electron transport chains in chloroplasts are very similar to those that we see in mitochondria. This figure illuminates the relationship between photosynthesis and cellular respiration. Do keep in mind that although plants fix all of the carbon by using energy from the sun, they still need to undergo the process of cellular respiration to break down the glucose that they've created in order to fuel the activities of their cells. Even though plants appear to be fairly static in their nature, within the cells of plants, they're very busy doing things, and every process requires the use of ATP. So we do not photosynthesize. We only break down products that are made by plants and other animals and digest them and release the energy in the form of ATP so that we can fuel all of our processes. However, plants will do photosynthesis in order to fix carbon and produce glucose. In addition, they need to utilize this glucose to make ATP. So they undergo cellular respiration to form all the ATP that they need in order to fuel the processes in their cells. Evolution does not necessarily result in optimum situations. Sometimes it just favors a workable situation derived from features that already exist, and photosynthesis is no exception to this concept. Rubisco, the enzyme that catalyzes the key carbon-fixing reactions in photosynthesis, provides a suboptimal solution. This enzyme has a second enzymatic activity that interferes with photosynthesis. That is, the active site on Rubisco has affinity for both oxygen and carbon dioxide. In addition to the carboxylation function that Rubisco has, adding carbon dioxide to RuBP, it also has an oxidation function, which we call photorespiration. This is where RuBP actually becomes oxidized by the addition of O2. So how does Rubisco know which to pick up? Carbon dioxide or oxygen? Well, the problem is that it doesn't. If there's lots of oxygen present, it'll pick up oxygen. If there's lots of carbon dioxide present, well, it'll pick up carbon dioxide. So the carboxylation function of Rubisco is favored under normal conditions. And the photorespiration function or oxidative function is favored when the stoma are closed or in hot conditions. So let's take a look here at the leaf. The leaf allows air and water to pass in and out of it through these pores called stomata, or stoma for singular. However, when it's very hot, the stomata will close. And as photosynthesis continues, we see a buildup of oxygen because oxygen is one of the byproducts. So the more photosynthesis that goes on while the stomata are closed will generate high oxygen conditions inside the cell. And we're consuming CO2, so the concentration of CO2 will decline. Thus, in hot, dry conditions when the stomata are closed, Rubisco is forced to pick up oxygen and oxidize RuBP, essentially undoing any carbon fixation that has happened. So photorespiration is an accident because Rubisco has two enzymatic activities. That is, its active site fits both carbon dioxide and oxygen, and its active site will pick up whichever one is most prevalent. The more oxygen there is trapped in the cell because of closed stomata, the more photorespiration those cells are forced to do. Now, there are a couple of solutions to this because obviously this is not a beneficial situation. We've already examined the C3 plant. This was where we were fixing carbon using the Calvin cycle and creating 3 carbon sugars for the first carbon fixation. So we fixed carbon onto a 5 carbon RuBP and instantly split it into 2 3 carbon sugars. This is a C3 cycle. There are 2 other types of photosynthesis, which are C4 and CAM, and both of these have evolved to overcome photorespiration. In both situations, carbon dioxide is fixed to PEP, which is otherwise known as phosphoenol pyruvate. You don't need to know that. PEP will be sufficient. But PEP is a 3 carbon molecule, and when we add carbon, it becomes a 4 carbon molecule. The enzyme that fixes carbon to become a 4 carbon molecule is called PEP carboxylase. PEP carboxylase has a much greater affinity to CO2, and thus it doesn't allow any oxidase activity. Basically, it traps the CO2 and holds onto it. There are two different solutions. First of all, there's a spatial solution, which we see in C4 plants. And then there's a temporal solution, both of which use PEP carboxylase to form a 4-carbon molecule. So even though one is called C4 and one is called CAM, both of them result in carbon dioxide being fixed to a 3-carbon molecule to form a 4-carbon molecule, hence the name C4 photosynthesis. Let's look at C4 plants. C4 plants are things like corn and sugarcane or sorghum and a number of other grasses that grow in hot, arid environments. And C4 plants separate carbon fixation and the Calvin cycle spatially. That is, they fix carbon to PEP using PEP carboxylase to form the molecule oxaloacetate. Here, we're going to carry the carbon into a different cell. In this other cell, a molecule called malate will be decarboxylated, and that carbon will be fed directly to rubisco. This way, rubisco is forced to pick up carbon dioxide instead of oxygen to maximize the output of photosynthesis. Now, you can know the name of the cells or not. We have mesophyll cells where the carbon is fixed and bundle sheath cells where the carbon is fed into the Calvin cycle. You don't need to know the names of those cells, just that in C4 photosynthesis, the fixation of carbon is spatially separated from the use of carbon in the Calvin cycle. Although this system overcomes the problems of photorespiration, it does have a cost. And that is, to produce a single glucose requires 12 additional ATP when compared to the Calvin cycle alone. C4 photosynthesis is advantageous in very hot and dry climates where photorespiration would remove more than half of the carbon fixed in the usual C3 pathway. Another solution is CAM plants. CAM plants are the succulents, water-storing plants, such as cacti and pineapples, and some members of about 24 other plant groups. In these plants, the stomata open during the night and close during the day. So there's temporal separation of the light reactions and the carbon-fixing reactions. This is reverse of what happens in most plants. So during the night, the stomata are open, and we're able to fix carbon. During the day, we capture energy from the sun and store it as ATP and NADPH. So CAM plants fix carbon using PEP carboxylase, just like C4 plants. They fix the carbon onto PEP during the night and store it in the vacuole. When the stomata are closed during the day to prevent loss of water, the organic acids are decarboxylated to yield high levels of CO2. So we've had CO2 stored in a very similar fashion that we see here. We fixed it to phosphoenolpyruvate to form oxaloacetate and then stored it. In CAM photosynthesis, we'll see the carbon dioxide is released when the stomata are closed to feed it to rubisco. This allows the plants to maintain high levels of CO2 and drive the Calvin cycle, thus minimizing photorespiration. So let's compare the both. CAM plants and C4 plants use both C3 and C4 pathways. In C4 plants, these two pathways are used in different cells. We do the carbon 4 fixation in one cell, and then feed that carbon to do carbon 3 fixation in another cell and run the Calvin cycle. In CAM plants, the C4 pathway happens at night in order to fix carbon and hold on to it until the day when the stomata are closed, at which point it releases it and undergoes C3 photosynthesis to produce sugars. So quickly to review, Photorespiration was the problem. And that results because Rubisco could pick up carbon dioxide or oxygen. If it picks up oxygen, that causes oxidation of RuBP, basically unfixation. So we ruin all the fixation that's happened for carbon. This is a problem. A few types of plants have adapted mechanisms to overcome this problem. Those mechanisms are C4 photosynthesis, where we separate the fixation of carbon spatially, and then CAM plants have adapted to separate the fixation of carbon in time. So at night we fix carbon in the C4 pathway. It's the same pathway. And in the daytime we release it into the Calvin cycle for C3 carbon fixation. So that brings us to the end of our chapter on photosynthesis. Let's take a quick card quiz. What structure is responsible for gas exchange? Making glucose via photosynthesis costs how many ATPs and how many NADPHs? However, 1 molecule of glucose is enough to make how many ATPs? So this one ties together both photosynthesis and respiration. What's the function of Rubisco? Where could a botanist expect to find C4 plants? The end product of photosynthesis is the starting material of cellular respiration. Is this true? Or blue, false? Which of the following prevent the formation of phosphoglycerate? That is PGA. Here are your answers. If you've got all of these answers, you're well on your way to a good understanding. But now it's time to really buckle down and work on your diagramming assignment as well as your Learn Smart modules in Connect.